classification and taxonomy of fungi. We'll see in detail about how this classification is actually started, what is taxonomy, why we need classification in fungi, and all those things. I'm N.S. Shankar Reddy, PhD plant pathology. So let's start with the basic definitions. What is classification? What is taxonomy? What is nomenclature? So what are the different kinds of governing bodies that is actually acting and undertaking this classification and all those things. So generally classification in the sense, so arrangement or aligning of units or organisms to the taxonomy group. Why this classification is required first of all? So when it comes to this in daily life also we used to classify many things. Even in the business sector also we used to classify many things for easy understanding or for easy studying or maybe for easy various kinds of things we used to classify, right? Here generally the classification is arrangement of a different units in the sense we can also consider it as organisms or maybe fungi or maybe bacteria or maybe virus or maybe various kinds of things generally. So why we are classifying for easy understanding or maybe for easy studying or for easy identification, various kinds of things we used to classify. Generally the classification in the sense arrangement of our classification of different kinds of units to the taxonomy group. Then what is this taxonomy? So there is a new word, right? So what is this taxonomy? So the taxonomy is generally a biological science which mainly deals with the study and the naming, classification, all those things that was dealt by a subject called taxonomy. The person who studies the taxonomy is called as, or who actually involved in this classifying these things are called taxonomists or systematists. So let me repeat once again. Taxonomy is generally a biological science. So however we have a plant pathology, it is also a biological science that mainly deals with the study of naming, classification, all those things, classification of organisms, all those things that was dealt by taxonomy. So nomenclature in the sense name, giving names, very simple. So nomenclature in the sense just giving the names, so according to their maybe characters or maybe various kinds of other things, we used to just give a specific name for that. So that was actually when it comes to fungi, every organism has their own governing body. For example, if you see fungi ICBN, International Code and Botanical Nomenclature, that was maintained by ICB. Then I can take viruses, ICTV or ICNV, ICTV now, because ICNV's name changed to ICTV, International Committee on Taxonomy of Virus, 1973, right? So this is general introduction about this taxonomy and classification. So why this classification is actually required? So in the beginning of mycology, I have given just a brief introduction about this mycology and where what are all the why we need to study mycology and all those things. So when it comes to the kingdom of fungi, it is a very diverse. So far, there are almost 2.2 to 3.8 million species of fungi have been identified. Among that, 120,000 to 130,000 species have been described so far. Described in the sense we know at least something about that fungi's fungal characters that was described. There are many fungi still yet to be described, right? So among that 120,000 species to 130,000 species of fungi, 8,000 species of fungi have been identified is plant pathogenic. Among that 300 species are human pathogenic. So just imagine how diverse it is, how just like very difficult to identify, right? So that's why he imagine based on their characters, maybe spore morphology, or maybe now we have advanced technologies based on DNA fingerprinting or based on their genetic structures or maybe various other kinds of things we may classify. So if we classify, that will be very easy to have a basic understanding or basic remembering the stuff or maybe basic various kinds of purposes. Now I will tell you why this exact classification is required. Is that much necessary, or why we exactly required, or why we exactly group this fungi into different classes or maybe phylum or genera or what other kinds of things are there. Why it is exactly required, I will tell you. So this fungal classification is generally required for to understand the evolutionary relationships. Evolutionary relationships, like where it is exactly what is the ancestor of the particular fungi, or like why it is, where it is actually evolved, something like that. So scientists use it to understand this evolutionary relationship. If we groups, or if we classify the organisms in a different phylums or other things. The second one is identification and study. It's a very important thing, like as I told you that there are plenty of fungi. If we cannot divide, or if we cannot classify, it will be like a very big mess. Even though you can take in a supermarkets also, they used to classify the things, clothings, or some other kinds of things. Just imagine if everything will be in a single place like a mess, it will be very difficult for the customer and consumer as well. Sorry, consumer and as well as marketing person as well, to identify where exactly the particular things will be there. So just imagine if we classify organisms or maybe various other kinds of things, especially let's talk about fungi because it's mycology, right? So fungi, it will be very easy to study, or to group the unknown fungi, or maybe compare the characteristics based on their maybe spore morphology. And scientists also, it will be very helpful if we identify our fungi that belongs to different groups for research, education, and as well as various other kinds of allied purposes, include agriculture and as well as industries. So the second one is a third one is a biodiversity conservation. So when it comes to biodiversity conservation, uh, for example, if there is an extinction fungi, like a fungus, something are going to extinct, imagine if you have a medical values, or maybe some other kinds of special kinds of medicinal properties. So by having this kind of classification, we can identify, and we can start to preserve it. And which means we can start protecting it, like endangered species, something will be there, we can protect it if having really medicinal importance and other various kinds of things. Then the next one is a medical application. Of course, we know very well about various kinds of fungi are actually used in the production of various kinds of medicines, like antibiotics, or immunosuppressants, or various kinds of medical compounds are actually produced from this fungi. The next one is agriculture and industrial importance, like production of various kinds of enzymes, beverages. You can take Saccharomyces cerevisiae, it will produce various kinds of enzymes, right? Uh, and various kinds of industries, they used to use like production of various kinds of beneficial substances that may be mainly used in industrial purposes. Biodiesel degradations and classification will definitely helpful, by using, and using in this very useful way, and just identifying the stuff, so which one is useful. So this agriculture and industrial applications. So the last one is environmental and ecological research. So the fungi generally plays a very crucial role in ecosystem, everyone will knows. So this will help scientists in various environmental process and their interactions with other organisms. So this is how why classification is required. So we have plenty of other kinds of purposes, right? So if we classify this fungi, it will be very easy to study, to identify the things used for various kinds of medicinal purposes, or agricultural purposes. Here we need to remember something here. Classification in the sense, don't go to the direct classification like Kirkatile classification, Ainsworth classification, Alexopoulos classification. Here generally classification is done on various bases. For example, if you see this based on the nutrition, so this is general introduction only. Of course, we have a detailed classification inside. So this is a general introduction based on the nutrition, how fungi is classified. So if you see, the plant kingdom is generally characterized by photosynthesis type of nutrition, right? So they produce their own food material by the process of photosynthesis. And when it comes to animal kingdom is ingestion nutrition, ingestion type of nutrition. So when it comes to the fungi, the most important thing is fungi, that is actually characterized by absorptive type of nutrition. They actually absorb the nutrients, so that through hyphae and other kinds of things, they used to absorb the nutrients. So this is absorptive type of nutrition generally. So based on the nutrition, this is general classification. This is not a detailed classification. You guys no need to worry about it. The next one is generally this various classifications are there. I'm giving some basic things here, that's it. So based on the different modes of nutrition, so this fungi generally classified different modes. Based on the nutrition, the fungi is classified. Based on the spore formation also, the fungi is classified. Now we'll see one by one. So first one is based on the nutrition in the sense food material, based on the nutritional requirement, how fungi is classified. So there are various other things also there, but I have placed only three important things that we generally use to study and generally use these kinds of terminologies or terms all the time. So the first one is saprophytic. I will tell you in a simple way rather than extending the things. Saprophyting means the fungi that grows or obtain the nutrition or get the nutrition from dead and decaying organic matters. Even after the death of the plant also they feed on them and they get the nutrition. For example, best example is rhizopus, penicillium, aspergillus. So these are all the best examples of saprophytes. So even after death of the tissues or even after the death of the plants also they will live on them, they will feed on them and they will get the nutrition. That is called saprophytic. Parasitic means the name itself indicates actually the fungi lives on the other organism and also cause the harmful effects to the other organism. For example, two organisms are there. So this fungi, it is a parasite which actually live on the expenses of other organism and it will get all the nutrients the parasite requires and also cause a harmful effect to the other organism which actually get helping to get the nutrients. So here generally the fungi obtain nutrition by living on the other living organisms and absorb nutrients on them and it causes harmful effects to the organism, that is called parasites. For example, you can take almost obligate parasites. So let me tell you this Taphrina, Puccinia, there are downy mildews. There are strictly obligate parasites. Okay. Next one is the symbiosis. Symbiosis means so mutual relationship or mutual beneficial relationship is called symbiosis. So like two organisms are there. So you can take an example of lichens and mycorrhiza. So one will provide the nutrition, another will provide uh, uh, shelter and all those things. Like both are benefited. This need a nutrition, that need a shelter. So examples I am telling. So here lichens and mycorrhizas are actually convert this solubilize this phosphorus, whereas the lichens will take that nutrition. It will provide the shelter to the mycorrhiza. So here this is actually a symbiotic relationship. No organism is harmful. No organism will get any harmful effect. Both are benefited, and such kind of relationships also there. For example, algae, fungi also we can also see, and here in an algae fungi a mutual benefit like fungi provide shelter for algae and in reverse algae synthesize the carbohydrates for fungi because fungi cannot produce their own food material, right, in such a way. So these are all the general modes of nutrition: saprophytic, parasitic and symbiotic. We'll see the next one. Next one is based on spore formation. So what kind of spores this group of fungi will produce? Generally, when it comes to mycology, we used to study four major phylum of fungi, that is Oomycota, Zygomycota, Ascomycota and Basidiomycota. So what kind of spores it will produce now we will see. Generally the division the classification is like a Chytidiomycota and Blastocladiomycota will produce a flagellate kind of cells. Zygomycota will produce zygospore with a suspensor. Glomeromycota generally endomycorrhizal fungi, that is vascular mycorrhizal fungi. Ascomycota will produce ascus with ascospores. So Basidiomycota will produce basidiospores, basidium with basidiospore. Here Oomycota generally produces oospore as a sexual spore, zoospore as a asexual spore. Here Zygomycota produces sporangia and planospores, sexual spore as a zygospore. Ascomycota and Basidiomycota produces conidia as a sexual spore, ascospore and basidiospore as a sexual spore respectively. So this is generally based on spore formation. These are all basic understandings and basic classification. So when it comes to original classification, we'll start with the history of original classification, how this fungi actually have their roots is for classification and all those things. So generally there is a man called Carolus van Linnaeus. He is generally considered as father of taxonomy. So he is the one first who laid the classification division and all the such kind of ideology was erected by this Carolus van Linnaeus only. After that, in the history of classification, if we dated back exactly at 18th century, until the one third of or until the first third of 19th century, there are golden findings, or there are various kinds of breakthrough contributions happens that is related to the fungal taxonomy and classification. So here CH Persoon in 1794, 1801, 1822, 28, he have done some contributions, breakthrough contributions to this fungal classification. Then Nies, Vasey, Isenbeck and E.M. Fries link those systematicians or the taxonomists have uh, classified fungi based on external characters, or based on morphology, based on the kind of fruiting bodies they produces, based on hymenophore, and based on the mode of spore formation. They have classified the fungi, because at the time we don't have they don't have sophisticated technologies. So they just by having the external characters or by having the morphological characters, so this is just classified the fungi. This is actually the origin of classification. So there are two important books which actually have a very important contributions that laid a very strong foundation for this classification, which includes Synopsis Methodica Fungorum, which was written by C.H. Persoon. He is a student of Carolus van Linnaeus, because Carolus van Linnaeus is considered as father of taxonomy, right? So this C.H. Persoon is a student of Carolus van Linnaeus, and the one more book, that is a System of Mycologicum with the additional index and Elenchus Fungorum. This was written by E.M. Fries. He is considered as a reconstructor of he is considered as a father of systemic mycology. So like these two books are very important contributions, like Synopsis Methodica Fungorum and System of Mycology, and these two books are that laid very strong foundation for this taxonomy and systematics. So their classification is generally based on the external characters, contemporary, like nomenclature and all those things. So this is general classification only, but these are all the very important books that laid initial strong foundation. So the next step in the classification after this CH Person and EM Fries is Silloge Fungorum, that was written by P.S. Secado. So P.S. Secado, he started classifying the fungi by in his book like Silloge Fungorum. So it's a very important book which actually comprises of almost 25 volumes, series of 25 volumes. Actually it's a 26, but probably one out of 26 one is not mentioning anywhere. So it's a 25 volumes of work where he have identified, classified various kinds of fungi. This is also very important next step when it comes to the fungal classification. So among the various publication, Secado and Snyder's work are basically regarding on the conidial stages, especially the fungi. They have deuteromycete, they have classified a lot. So such kind of things they have done. And when it comes to the subsequent studies like Ainsworth classification, Moore classification 1971, so they introduced a new thing called deuteromycotina or deuteromycetes fungi, which is actually a conidial fungi at that time. So this fungi actually produces no sexual stages, or no sexual life cycle can be observed. So then I will start with the detailed one. So the first one is the Secardos classification. So in 1866 he have divided the true fungi into 4 classes. So the first one is the Picomycetes, Ascomycetes, Basidiomycetes, and another class is Deuteromycetes. So these are all the fungi. Out of the fungi, so generally he generally classified based on the presence of septa, or the maybe characteristics of production of the sexual spores like oospore, zygospore, ascospore, and basidiospore. Based on that characters, he actually classified. And he, out of the fungi, 4 classes of fungi he have described: Ascomycota, Basidiomycota, and Deuteromycota are higher classes of fungi, or higher fungi, whereas Picomycetes he actually grouped called as a lower fungi, that actually belonging to the lower group. That's why it's called as a lower fungi. That is his classification, or his ideology about the classification. When it comes to the next one, further Aloxopolus classification, this is also widely accepted and popular classification. So this is generally a 9 classes will be there in this Aloxopolus classification. So the Aloxopolus put actually the division Mycota into 2 subdivisions. Generally Mycota into 2 subdivisions. Myxomycotina or Myxomycetes, Eumycota are true fungi he classified. So the 9 classes of Eumycota fungi are Chytridiomycetes, Hypochytridiomycetes, Plasmodioporomycetes, Oomycetes. All the earlier fungi of belonging to this Phycomycetes are just he placed into the various other groups like Hypomycetes, Plasmodioporomycetes, Oomycetes. And the next one is Lygomycetes, Rhynchomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Generally, Deuteromycetes are fungi imperfecti uh, because they lack sexual stages. This is the actual 9 classes classification that was given by Alexopoulos and Mintz. This is also very famous classification. So the next one is this man have done really a breakthrough Ainsworth classification. So very important. Still few people are following this classification as well. He divided into 2 divisions, Mycota and Eumycota. Mycota is a plasmodial forms. Eumycota is actually a non-plasmodial form, which was actually the true fungi. So the true fungi were again divided into 5 subdivisions: Masticomycotina, uh, Lygomycotina, Ascomycotina, Basidiomycotina, and Deuteromycotina. This is the Ainsworth classification. So again, the Ainsworth further improved and further improved the same previous classification. This is also very important classification, and many textbooks are following the same Ainsworth classification only. He divided the fungi into 2: Plasmodium present, Plasmodium absent. That is Plasmodium present like a Myxomycota group of fungi, true slime molds, whereas Plasmodium is absent, Eumycota true fungi. So like when it comes to Myxomycotina, Crassiomycotina, Hypomyxomycetes, Myxomycetes, Plasmodioporomycetes, that he placed under this Myxomycetes. So when it comes to this Oomycota as a true fungi like Masticomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, Deuteromycotina. Generally Masticomycotina water molds are comes under like Chytridiomycota, Hypochytridiomycota, and Oomycetes. So when it comes to Zygomycota, Zygomycetes and Trichomycetes. Ascomycota, Hemiascomycetes, Plectomycetes, Pyranomycetes, Discomycetes, so Labyrinthodontomycetes, Loculoascomycetes, these all comes under Ascomycota. From the Basidiomycota, Teleomycetes, Hymenomycetes, and Gastromycetes. And Deuteromycota, there are imperfect fungi, Blastomycetes, Hypomycetes, and Coelomycetes, these all comes under. So generally, this Zygomycota are conjugate fungi because this male and female just combined to form zygote, right? Ascomycota is called sac fungi because it produces sac-like structure. It is called a club fungi where the basidospores are produced above the club, right? Deuteromycota generally called is imperfect because it produces no sexual stages. And based on the what kind of spores it will produces, here I'll mention generally this Masticomycota water molds produces oospore as a sexual spore, zoospore as a asexual spore. When it comes to Zygomycota, aplanospores, sorry, before I told that planospores, right? No, it's aplanospore. Non-motile spores are called aplanospores, and zygospores are the sexual spores. When it comes to Ascomycota, ascospores are sexual spore. Basidiomycota, basidospores, and Conidia both commonly produces. So this is the Ainsworth classification. This is also very famous classification. Coming to the next classification, Alexopoulos and Mims classification that was given by 1979 by Alexopoulos and Mims. So they divided the mycetes fungi into three divisions: Gymnomycota, Masticomycota, and Amasticomycota. So this Acrasiomycota, Plasmodioporomycota, all those things are comes under Gymnomycota. When it comes to Masticomycota, now again 2 divisions. So Amasticomycota, Zygomycota, Ascomycota, Basidiomycota, and Deuteromycota. He placed under this Amasticomycota. Again, this classes are given here. So this is Alexopoulos and Mims classification. This is also one of the important one. Now, when it comes to the present classification, the most important classification, and many scientists are following and many one are studying. For example, if you see, even in UG nowadays in UG courses and various kinds of master's courses, the present system of classification that what we are following is a Kirk et al. Classification. So this Kirk et al. Classification is the widely and the most accepted and widely regarded and following classification for the classification of fungi. It is actually that given in the 10th dictionary of fungi in 2008. So this dictionary of fungi, dictionary of chromistones, staminophiles, fungi like organisms, dictionary of protozoans and organisms, all put together, he came up with a very solid classification. That is the present classification, Kirk et al. It is the 10th classification of fungi, which means before 9 systematicians have done the classification. This is the 10th classification of fungi, which was published by Kirk et al. In 2008. So before also other classifications are like there, like Dick classification 2001. The same Kirk et al. has also done the classification in 2001, but that was further improved by the same Crick in 2008. So this Crick et al. Classification or 2008 classification is largely based on AFTAL. AFTAL means a project called Assembling Fungal Tree of Life, where all the sequence analysis or multigene sequence analysis was undertaken, and where these are classified. So this after all project was actually started. Assembling Fungal Tree of Life was actually started in 2001. That was funded by the National Science Foundation in USA, and more than 100 collaborations from 20 countries was involved in. So this project actually enhances the evolutionary understanding about the organisms, especially the fungi, because it is a very oldest class from species that was living on Earth, right? So to have a detailed structural understanding, and we have evolutionary organisms or relationships, so this project will help. So the outline classification again divided into three groups and 8 recognized phyla in the kingdom fungi. So he mainly concentrated on especially fungi only. So this is generally I placed for our understanding only, sir, what kind of spores that is produced. General understanding only. There is nothing to confuse too much here, because that I have already explained about it, right? So this is general classification of Crick et al. 2008. So Protozoa, Chromista. Again, this fungi 8 classes are there. That is the most important one here divided. I think I have given only 7 here. One more thing also there. Chytridiomycota, Neocalibastigomycotina, Cladomycotina, Glomeromycotina, Zygomycotina, Ascomycotina, and Basidiomycotina. I think is Deuteromycotina is also there in this, but now Deuteromycotina was actually placed in the Ascomycotina. One more classification is there, I think so. Now we'll start with the taxonomy, since we already discussed this is a biological science which mainly deals with the naming and classification of organisms. So now we will see. So let's start with this careless swan lineage. Before going, what are the different kinds of governing bodies that regulates this classification? So generally, this careless swan lineage is considered as a father of taxonomy. I introduced a system called binomial nomenclature. So binomial means every organism has two names, genus name and species name. That is called binomial nomenclature. So it's a very important one and a very chief starting point or foundation for the naming and classification of fungi. He grouped living things into hierarchical categories, and 7 level of classification was also introduced by Carolus van Linnaeus. 7 level of classification is nothing but classes, orders, genus, species. This is the 7 level of classification, nothing to worry about it. So this is actually he have given a lots of breakthrough foundations for this classification. That's why it is considered he is considered father of taxonomy. Now we'll see how this actually the taxonomy is the roots. So the taxonomy is generally describe and classify the organisms based on the knowledge, understanding, and various kinds of things. Here, Carolus van Linnaeus is considered for the father of taxonomy, and he introduced binomial nomenclature and naming of species based on his books. He wrote two famous books, Species Plantarum, where he have described around 5,900 species of plants, and Systema Nature, where he have described around 4,200 species of animals. From this Species Plantarum, he erected a concept called binomial system. There he started naming the fungi, or he started dividing the fungi into two names, genus name and species name. That is the chief starting point. So what are the principles, rules, and regulations? This botanical nomenclature is independent to the zoological and bacteriological and viral nomenclature. So coming to the animals, bacteria, and virus, this is completely independent nomenclature. So there they followed a little bit different system. Of course, more or less similar only, but a little bit different system can be followed. But this binomial nomenclature is independent of this zoology, bacteria, and viruses. So generally, as I told you, this species plantarum has laid the this publication laid the chief starting point. So to avoid the undesirable effects, uh, again he divided into a hierarchy like families, genus, species. It was the names was given to this particular taxas like families, genus, and all those things, and this binomial nomenclature is widely accepted all over the world, and it is a very famous one. Nowadays, still we are following it, right? It's been almost 200 years since the discovery, almost 180 years since the original foundation. Still, we are using this binomial nomenclature in all kinds of things. So this presently governing bodies includes so algae and fungi and plants was taken care by International Code and Nomenclature of fungi, plants and fungi, algae, fungi and plants. So the name was actually replaced to ICBN, International Code and Botanical Nomenclature, and either International Rules of Botanical Nomenclature. So they will provide the rules and regulations, various kinds of things. This is the governing body. So for animals, International Code on Zoological Nomenclature (ICZN). When it comes to bacteria and archaea, International Code on Nomenclature of Prokaryotes (ICNP). In 2008, it is replaced to International Code on Nomenclature of Bacteria. For viruses, ICNV, International Code on Virus Classification and Nomenclature, that is name changed to ICTV, International Code on Taxonomy of Viruses in 1973. So these are all the various governing bodies that they are actually working on this taxonomy and nomenclature, and they are all the various kinds of things. Now we'll see what are the different kinds of rules for writing the scientific names. So first starts with the binomial system. So the name itself indicates two names. First one is a genus name, second one is a species name. This binomial nomenclature was actually given by Carolus van Linnaeus that we discussed earlier. So the genus name have actually no fixed endings. For example, we can take Puccinia graminis. Puccinia is a genus name, graminis is a species name. So the next one is a trinomial system or trinomial nomenclature, which means three names. So adding the third name to the binomial, which means so adding the third name to the binomial that provides the further information about the organism, or the further information maybe the form of species or races or pathovars and all those things. For example, Puccinia graminis, forma species, Tretici. Puccinia is a genus name, graminis is a species name, forma species, Tretici. This is a trinomial system of nomenclature. And one more rules or system will be there. So at the end we add the author names. For example, name of the taxonomist who actually first identified it or who actually first discovered it. We used to give the abbreviation of the scientist name. For example, we can take Fusarium oxysporum forma species vas infectum. So GF Atkinson, we have given a shortcut name GF80K, double C Snyder and HN Hansno. So here just like giving shortcut names that we are ending at the end of the this trinomial system or binomial system that gives provide the further information about the author also, that who actually discovered this organism. Actually, if you see these abbreviations and giving the author names that we can mostly observe in the Ainsworth and Bisbee dictionary of fungi, where they have mentioned very clearly about every single fungi. They have mentioned at the end author name. This is called author name, right? So mostly we are using a binomial system, we can see everywhere, but sometimes you can also encounter with the trinomial nomenclature. There is nothing much variation about binomial and trinomial. So in binomial, two names will be there, genus and species name. In trinomial nomenclature, one more name, like maybe form of species or races, that provides the extra information about the organism as well. That is trinomial nomenclature. So now we will see the important guidelines for the nomenclature. What are all the guidelines that must follow? Generally, these binomials are usually derived from Greek or Latin, either Greek or Latin, and the genus name must be capitalized. These are all the very important role that we must follow for giving our naming of any organism. So genus name always starts with a capital letter, while rest including the species name and other letters or other subspecies name, everything will be small letters. For example, we can take Puccinia graminis. So the Puccinia starts with a P capital letter, right? Genus always starts with a capital letter. Graminis always, like, except the genus, the remaining all things will be as usual like a small, normal common letters only. The leaving, there is a single digit gap between the genus name and the species name. If we see here, for genus name and species name, there is a single digit gap that actually gives clarity about for reading and all those things. So giving a single digit gap between genus name and species name. And in case of where author name is including, use standard abbreviations of the author name. For example, Atkinson ATK. For example, Anton D Barry or Barry. Generally D Barry can be used, or maybe EF Smith, generally Smith, like that. A short kind of terminologies or abbreviations can be given generally. The next one, next rules are binomials when are handwritten that must be underlined. Actually, it not supposed to be in italics. Actually, if you are writing in handwritten, we cannot able to write exactly as like a computer in italics, right? So that if you are writing in a handwritten way, we must underline genus name and species name individually. When it is a printed one, we must italicize it. There is no need to underline it, because in printing we have an option called italics, right? So that we can italicize there itself. So there, in such kind of cases, we no need to underline it. But if you are writing in a handwritten way, we must underline the genus name and species name separately. And the taxa is actually classified into a hierarchical classification. That is, taxa is classified into kingdom, division, class, order, family, genus and species. These are all the hierarchical classification, right? Again, this division is divided into subdivision. Again, this class is divided into subclass. Order is divided into suborder. So these are all the general hierarchy or taxonomy system of tax of classification. Here, genera and species have actually no endings. There is no specific endings to this genera and species. And sometimes it may be broken into species, may be broken into variety or form of species, that may be observed in many cases, right? So this is actually the 7 level of hierarchical classification. Starts with kingdom, phylum, class, order, family, genus, species. Any organism, any classification you take, this is the hierarchical classification. This is the suffixes or phylums or categories and all the taxons that was given by ICN, that is International Committee on Nomenclature of this fungi, especially fungi and fungi-like organisms. So this is the hierarchical classification. Always starts with the kingdom, phylum, class, order, family, genus, and species. So here, one thing we just have a little brief introduction about species. Generally, these are a group of individuals which actually resembles their morphological and reproductive characters, and they also have capability to produce the offsprings. Generally, we can call it as a unit, like a species. For example, we are belonging to the human species. They are belonging to the animal species. Some are insect species, like that. A separate division, a separate group. So that is generally called as a species. So here, according to this ICBN International Code on Botanical Nomenclature, there are suffixes and phylums. There are standard endings. Always, they are always standard endings. Here, phylum always ends with the mycota. Subphylum always ends with the mycotina. Class always ends with the mycetes. Subclass always ends with the mycetidae. Order always ends with the ales. Family always ends with the ACA. This is whatever the classification, whatever the fungi it is. So this is the standard endings. For example, we can take an example of a wheat stem rust. For example, phylum comes under Basidiomycota. Here, mycota, we can see here. Mycota always ends with the mycota. Subphylum Basidiomycotina. Mycotina always ends with the mycotina, right? The class is Puccinio-mycetes. We can see here mycetes. The next one is the subclass Puccinio-mycetidae. Actually, subclass always ends with the mycetidae. You can see here. Order always ends with the ales, Pucciniales, ales. Family always ends with the ACA. We can see family Pucciniaceae generally, right? So this is the standard endings for you can take any fungi, you can take any organism that comes under classified that is actually follows the rules and regulations of ICBN. These are all the standard endings. It's a very important in considering the net exam as well as the various other exams also. They will ask like what is the suffixes or endings, the standard endings of phylum or subphylum or class or subclass or order or family. So like this they may ask, okay, phylum always ends with mycota, subphylum always ends with mycotina, family always ends, class always ends with mycetes, subclass always ends with mycetidae, order ends with ales, family ends with ACA. And grouping, these are all the various kinds of famous taxonomies that they have done the classification. These are all very famous taxonomies. So which starts with the Linnaeus. Linnaeus actually gave two kingdom classification, that is a Plantae and Animalia. Actually he divided entire kingdom into two kingdoms. Plants, entire plants are he placed under plant kingdom, entire animals he placed under animal kingdom. Again, Ernest Haeckel in 1866 he erected a three kingdom classification. To the two kingdom classification that was given by the Linnaeus, he added extra thing that is called Protista. Plantae and Animalia, whatever the Linnaeus gave, the same things will be there. He added extra things, so Protista. Okay, so where he actually put all the bacteria and all those things into. So later, Copeland, he erected a four kingdom classification. Whatever the Haeckel gave three kingdom, like Protista, Plantae, Animalia, three are there. He added extra things, Monera, where whatever this Haeckel, where he placed all the bacteria into this Protista. But when it comes to Copeland, he slightly divided. He placed the prokaryotes into Monera. Protista only contains eukaryotes, as a plant and animal as common plants and animals. And five kingdom classification, it's a very important. It was given by Whittaker classification. This is one of the most important, widely regarded classification in the sense just kingdom divisions, not that's it, nothing else. So in exams also, five kingdom classification was given by Whittaker. So such kind of questions I have already saw. So five kingdom classification, when it comes to here, Monera, where he placed Monera, Protista, Plantae and Animalia was there, which was the same things that Copeland also gave. But the special thing the Whittaker have done is he gave a new kingdom, that is a fungi. For entire fungi he gave a separate kingdom. All the fungi he placed in the kingdom called fungi. So that's why this Whittaker classification is very important. So five kingdom classification: Monera, Protista, fungi, plant and Animalia. So Carl Lewis in 1977 he gave six kingdom classification. So where he divided a eubacteria and archaebacteria into two: Protista, fungi, plant and Animalia are common. The Chatter 1937 he gave two empires: eukaryota and prokaryota. Carl Lewis, the same Carl Lewis, given three domain classification: bacteria, archaea and eukarya. So these are all the general taxonomies or systematics of what actually just classified the organisms. Generally, Pliny the Elder, who introduced the first artificial system of classification in his book Historia Naturalis, and it's a very old book, it's almost 2000 years old, right? All right. So, and then Swinch botanist Pyrrhus de Candolle actually coined the term taxonomy. It's a science actually naming and classification of organisms. And Lamarck and Treveris coined the term biology. So this is general, just some kind of inputs, nothing else. And this is the classification that we are going to follow for now. Of course, Alexopoulos classification, Mims classification, and Whittaker's classification, sorry, other kinds of classification also there. But in this entire mycology, I am going to follow this present classification, that is a Kirketal classification, 2008 Kirketal classification, that is the most advanced. Of course, after Kirketal also, Heidi et al. 2021, various other kinds of classification also came out, but that was, those classifications are not that much famous. But this classification, since it is based on the Aftal assembling fungal tree of life, this classification was very famous. Now the entire mycology or upcoming videos are, I am going to follow the same kind of classification with slight variations. For adding the extra information, I may follow maybe Ainsworth classification or maybe Alexopoulos classification, but 99% of the cases I am going to follow the same classification what I mentioned here: Protozoa, Oomycota, and the separate kingdom fungi: Chytridiomycota, Neocalisticomycota, Blastomycota, Glomeromycota, Zygomycota, Ascomycota, and all those things that we are going to discuss. And this is the classification that I am going to follow. So for further information, students can refer my book, A Vision into Plant Pathology, complete student version. For further doubts and clarifications, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.